What happens when memory immune cells forget their memories? Without memory of past infections, our immune system is less able to fight off pathogens. The problem is that sometimes pathogens themselves are responsible for this so-called immune amnesia. Measles virus, an incredibly contagious virus that is currently spreading in the US and other world regions, can trigger immune amnesia. This makes people with measles vulnerable to secondary infections even after the measles infection has resolved. To that end, scientists are exploring treatment paradigms to prevent measles-induced immune suppression and mitigate severe infection outcomes. Welcome to Microbial Minutes, the American Society for Microbiology's update on what's hot in the microbial sciences. I'm Madeline Barron, Science Communication Specialist at ASM. Before we dive into today's video, please take a moment to fill out the survey linked in the description. It's 5 questions, takes less than 3 minutes, and your answers will help us make better videos. We appreciate the support. To begin, let's first cover some basics about measles. The disease is caused by an RNA virus belonging to the Paramyxoviridae family of the morbillivirus genus. The virus, of which humans are the only natural host, is spread through respiratory secretions and is extremely transmissible. 90% of people who don't have prior immunity to measles via vaccination or previous infection will develop disease if exposed. Symptoms of measles include a high fever, cough, and about 14 days post-exposure, a characteristic rash that begins near the hairline and face and blooms across the rest of the body. The disease is associated with children, though anyone can get it. Measles can be serious, leading to long-term and potentially fatal outcomes. The good news is that vaccination is incredibly effective at preventing measles. 2 doses of the measles vaccine, widely given as part of the measles, mumps, and rubella, or MMR vaccine, is 97% effective at preventing measles. However, ongoing measles outbreaks persist globally. Declining vaccination rates, fueled in part by the COVID-19 pandemic and a rise in vaccine hesitancy, has increased the risk for measles outbreaks, including in areas, like the US, where the disease had previously been eliminated. Moreover, measles is not the only respiratory disease that is spreading, particularly at this time of year. Others, like COVID-19, flu, and RSV, are also making the rounds, though the relationship between measles and these unrelated respiratory viruses on health outcomes is not clear. One of the key problems with measles is that it's more than a fever and a rash. It can also cause immune amnesia, which, as mentioned up top, is when the immune system forgets how to fight pathogens. During infection, the measles virus infects memory T and B cells, which spread throughout the body. Clearance of infection means that these cells must be destroyed, reducing the pool of memory cells in the host. Notably, memory T and B cells produced after a measles infection only remember the measles virus. Most other pre-existing immunity has been erased. This leaves patients vulnerable to secondary bacterial and viral infections, and it can take 2 to 3 years for memory to be fully restored. Better understanding of the timeline of this immune decline and strategies to prevent it would be ideal, particularly as risk of measles outbreaks continues to rise. To that end, in a recent study published in Nature Communications, scientists sought to gain experimental insight into the time window by which measles virus-induced immune suppression could be prevented through direct-acting antivirals, that is, compounds that inhibit viral enzymes needed for replication. None of these compounds are currently available to treat measles. Rather, management of measles is primarily supportive care. For unvaccinated individuals or those with no pre-existing immunity, prophylactic administration of the MMR vaccine within 72 hours post-exposure to the measles virus may offer some protection against infection or severe disease, though it seems to be most effective in limited contact settings. Less so if an individual had intense, prolonged, and close contact with someone with measles. Immunoglobulin, derived from blood plasma from thousands of donors, provides antibodies that can also prevent or modify the measles disease course in people without immunity, if administered 6 days post-exposure, though this is limited to certain cases and in high-income countries. Last year, researchers did investigate whether remdesivir, a broad-spectrum antiviral drug known for its use for treating COVID-19, was efficacious as a post-exposure prophylaxis for measles and rhesus macaques. The results weren't promising, however, showing that while the drug transiently suppressed expression of measles virus RNA and eliminated dissemination in the body, the virus was not cleared and there was no improvement in clinical disease. In all cases, the specific impact of post-exposure prophylaxis on virus-induced immune suppression was not entirely clear. Which brings us back to today's study. Here, scientists used a ferret model of disease in which the animals are infected with a recombinant strain of canine distemper virus, or CDV, a morbillivirus like measles. This is a model for measles virus-like disease, and the scientists found strongly supports the morbillivirus immune amnesia hypothesis. Prior to infection with CDV, animals were vaccinated against influenza. The idea was to create pre-existing immunity to a virus so that later the researchers could assess if and how that immunity changed post-CDV infection. The ferrets were then infected with CDV 2 weeks after their influenza booster dose. Following CDV infection, the ferrets were orally administered a broad-spectrum inhibitor of paramyxovirus polymerase, which is required for viral replication. The ferrets received the inhibitor, known as GHP 88309, at various points post-infection, which corresponded to distinct stages of disease, shown here. In other experiments, the scientists had found that treatment up to 5 days post-infection promoted complete survival of animals and helped mitigate loss of lymphocytes in the blood. There was also a vehicle group and a group of animals that had been vaccinated against CDV prior to the experiment. 5 months after infection with CDV, the researchers examined the immune response to infection with H1N1 and H3N2 influenza viruses, as well as CDV. Treatment with the broad-spectrum antiviral at the first signs of clinical disease, in this model, 5 days post-infection when fever first appears, preserved titers of neutralizing antibodies against influenza viruses, indicating that pre-existing immunity was preserved. This was also true for rabies virus, which the animals had pre-existing immunity to as well. Results were similar as those that had been vaccinated against CDV. These findings, among others not discussed here, indicate that treatment with a direct-acting antiviral inhibitor when symptoms of morbillivirus disease first emerge fully protected against virus-induced immune suppression. It's worth emphasizing that this paper used an animal model of measles-like disease, and it's not exactly the same as measles disease progression. Moreover, the compound, GHP 88309, cannot be used in humans. This wasn't a study focused on finding a drug for measles. However, what these findings do establish is a paradigm for morbillivirus treatment. That is, they show how therapeutic mitigation of primary clinical signs of morbillivirus disease and immune amnesia is beneficial when treatment is initiated before or at peak of primary viremia. Of course, in the context of human disease, this requires identifying efficacious direct antiviral compounds and further investigating the dynamics of disease progression and immune suppression. But this study provides a foundation for doing both of those things. Now, there's another piece of this study that bears mentioning as well. As I highlighted earlier, there are more viruses than measles circulating in the population. How might these other viruses influence outcomes of morbillivirus infection? To find out, the researchers inoculated influenza-naive ferrets with a non-lethal influenza virus, then, 4 weeks later, infected with non-lethal CDV. Strikingly, animals consecutively infected with influenza and CDV developed severe hemorrhagic pneumonia with spontaneous bleeding, and they ultimately died. Those only infected with CDV survived. Similar results were observed when animals were infected with RSV first and then CDV. The study authors hypothesized this is because CDV infection during or shortly after recovery from prior infection alters quality of the host response to morbillivirus disease. Treatment with GHP 88309, 5 and 7 days post-infection with CDV prevented this severe disease from happening. So again, this was a model and does not directly translate to human disease. However, this facet of the study demonstrates how other viral infections can predispose to severe morbillivirus disease outcomes. They suggest that understanding a patient's recent infection history can be important for disease severity and progression, and how to manage it. This is useful given we are living in a time where measles cases may increase and are increasing amidst other viral threats. If anything, the results of the study emphasize the importance of taking steps to avoid measles infection altogether. How? Well, vaccines are a great place to start. That's all for today, and don't forget to take the survey linked in the description. I want to thank you for listening, thank Ray Ortega for production, and I'll talk to you soon.